Good day, everyone. I am Rosita Mabolo, and my research study is entitled Microbiological Analysis and Coliform Qualitative Examination on the Deep Well Drinking Water Source of Barangay Pinatilan, Valencia City, Bukidnon. So for the background of the study, groundwater is heavily used all over the world as the primary source of domestic drinking water supplies, and contaminated groundwater certainly enhances risk to public health. Water safety and quality are fundamental to human development and well-being. Providing access to safe water is one of the most effective instruments in promoting health and reducing poverty. Safe and readily available water is important for public health, whether it is used for drinking, domestic use, food production, or recreational purposes. Improved water supply and sanitation and better management of water resources can boost countries' economic growth and can contribute greatly to poverty reduction. Contaminated water and poor sanitation are linked to transmission of diseases such as cholera, diarrhea, dysentery, hepatitis A, typhoid, and polio. Absent, inadequate, or inappropriately managed water and sanitation services expose individuals to preventable health risks. This is particularly the case in healthcare facilities where both patients and staff are placed at additional risk of infection and disease when water, sanitation, and hygiene services are lacking. The pollution of drinking water is responsible for a large number of mortalities and morbidities due to waterborne diseases like protozoan and helminthus infections. Several studies have shown that microbial pathogens such as Salmonella, E. coli, S. faecalis, and enteroviruses are relatively stable in groundwater. Moreover, pathogen contamination, example are bacteria, protozoa, and viruses, poses a serious risk in water resources. The transport of pathogens from surface water to groundwater increases the vulnerability of groundwater. Barangay Pinatilan of Valencia City, Bukidnon supply drinking water to other three barangay nearby. These are Barangay Katumbalon, Mabuhay, and Tungan-Tungan. This water source is pumped from a deep well specifically in Purok 9 in the said barangay. Water issue in the community still continue to be an enormous. Development and growth put stress on the water sources and water quality. For the objectives of this study, the study's aim is to determine the quality of drinking water and the antibiotic resistance of coliform bacteria isolated from the deep well source in Barangay Pinatilan, Valencia City, Bukidnon. Specifically, it aims to determine the number of coliform, most probable number, per 100 ml of the water samples. Second is to identify the coliforms in the water samples from the deep well source, and last, to assess antibiotic resistance of the isolates to two antibiotic agents, the streptomycin and amoxicillin. For the significance of this study will evaluate the safety of deep well water for drinking and other uses in Barangay Pinatilan, Valencia City. The data that we'll gather during the assessment can be used to supplement the existing water quality status with data from other quality assessment studies carried out by the local government unit of the city. This research would also disclose details about coliform isolates antibiotic resistance. Furthermore, the findings of this study may be used as a foundation for the future research. For the limitation of this study, the study area that will cover is limited only to the pump station 12 of the barangay. The biochemical test will be focused only to the identification of E. coli and Citrobacter pyogenes and did not include other waterborne pathogenic microorganisms. The analysis will not include other standard methods for assessing water quality, such as single-factor assessment, water quality grading, comprehensive pollution index, and heterotrophic plate count system. Furthermore, for the biotic susceptibility test, Only two common antibiotic agents will be used. These are streptomycin and amoxicillin, which are both available in pharmacies. So for methodology, we have first the place and duration of the study. So the local of the study will be at Pinatilan, Valencia City, Bukidnon, specifically at the pump station 12 of this barangay. And then the time span will be 2 months right after the approval of the proposal. And then we have now the general experimental approach. So to begin with this, we should have first the an entry protocol. An entry protocol will be submitted to the barangay captain of this barangay as a formal letter of communication for the approval of the sample collection. So for sampling procedures, the water sources that will cover the water sampling included tap water lines that split separately to each destination barangay nearby, which are all nearest from the main pump station site. In each tap water line, three sampling stations will be initiated for replication purposes of the samples. The water samples will be collected with the use of proper sterilizing technique. And in this case, the sterilized glass bottle will be used to collect the samples. The opening of the faucet should be wiped to remove any adhering dirt before collecting water samples, and they should be opened to their maximum rate, allowing the water to run for 2 minutes. Plastic faucets should be cleaned and disinfected with alcohol. After several minutes of running water, 100 ml of water will be collected and leaving enough space of the sampling bottle. The collected water samples will be then immediately placed in a light-proof insulated box containing melting ice or ice packs with water to ensure rapid cooling. If ice is not available, the transportation time must not exceed 2 hours. The samples should be kept in the dark and that cooling is rapid. After the sampling procedure, multiple tube fermentation technique will follow, and then the MPN analysis, and then the indole, methyl red, Voges-Proskauer, citrate, and catalase test will also follow. So in multiple tube fermentation technique, we have 3 tests. First, we have coliform group presumptive test for coliform organism. The presumptive test is a screening test to sample water for the presence of coliform organisms. If the presumptive test is negative, no further testing is performed, and then the water sample or the water source is considered microbiologically safe. If, however, any tube in the series show acid and gas, the water is considered unsafe, and the confirmed test is performed on the tube displaying positive reaction. So in this test, water sample will be shaken 25 times through an arc of approximately 1 foot in a time limit not exceeding 7 seconds. By using a sterile 10 ml pipette, transfer 10 ml water sample to each of the large lactose fermentation tubes containing 10 ml of double-strength lactose broth. Inoculate lactose fermentation tubes at 34 degrees for 24 to 48 hours will be then incubate. So second, we have the confirmatory test. Confirmatory test, divide 1 or 2 using methylene blue agar plate into sectors and streak into each a loopful of inoculum from a positive presumptive lactose broth tube. Incubate at 34 degrees for 24 hours. After incubation, examine the using methylene blue plates with presence of colonies of coliform bacteria which are purple-red in color, 2 to 5 millimeter in diameter, and with a characteristic metallic sheen with a viewed when reflected light. And then we have the complete test. So biochemical test will be the complete test for the ultimate identification of E. coli. For each replicate, one representative EMB plate with green screen colonies from each replicate will be picked for pure culture. While isolated green metallic sheen or colony that will produce will be transferred to a nutrient agar plate as a working culture for gram staining, and biochemical tests will be done in duplicates. And then the MPN analysis, the number of positive tests in lactose broth of each replicate will be recorded and consolidated in the standard MPN index to determine the MPN of total coliform of each replicate. And then now we have the IMVC biochemical test. So first procedure or first test, the indole test. Production of indole is detected using Ehrlich reagents or Kovacs reagents. Indole reacts with the aldehyde in the reagent to give a red color. An alcoholic layer concentrates the red color as a ring at the top. And then the second test will be the methyl red test and the Voges-Proskauer test. This is to detect the ability of an organism to produce and maintain stable acid end products from glucose fermentation. Some bacteria produce large amounts of acids from glucose fermentation that they overcome the buffering action of the system. Methyl red is a pH indicator, which remains red in color at a pH of 4.4 or less, while MR test is useful in detecting mixed acid producers. A VP test detects butylene glycol producers. And citrate utilization test. This test detects the ability of an organism to utilize citrate as a sole source of carbon energy. Bacteria are inoculated on a medium containing sodium citrate and a pH indicator bromothiamine blue. The medium also contains inorganic ammonium salts, which is utilized as sole source of nitrogen. And then we have the catalase test. The catalase test tests for the presence of catalase, an enzyme that breaks down the harmful substance hydrogen peroxide into water and oxygen. If an organism can produce catalase, it will produce bubbles of oxygen when hydrogen peroxide is added to it. So as continuation, other biochemical tests will follow after the MVIC test, and then antibiotic susceptibility screening, then reading of well plates by microplate reader, then decontamination. So in other biochemical tests, we have 5 different tests under this. So the first one, we have the triple sugar iron fermentation test. So the triple sugar iron fermentation test is designed to differentiate among the different groups of genera of the Enterobacteriaceae, which are all gram-negative bacilli capable of fermenting glucose with the production of acid, and to distinguish them from other gram-negative intestinal bacilli. Then second, we have the gelatin hydrolysis test. Is a biochemical test performed as a presumptive test for the identification of Staphylococcus, Enterococcus, and other gram-positive bacilli. The objective of this test is to test the organism's ability to liquefy gelatin by the production of gelatinase enzyme, and then to differentiate organisms into different groups based on their ability to hydrolyze gelatin. And then mannitol salt agar test is used to determine organisms capable of using mannitol as food source and will produce acidic byproducts of fermentation that will lower the pH of the media. And then Salmonella and Shigella test used to differentiate Salmonella from Shigella. Salmonella is a rod-shaped microbe. Shigella is a slender-shaped microbe. Salmonella is a motile with peritrichous flagella, and then Shigella is non-motile in nature. Then the growth in potassium cyanide test in broth. So this test determines whether the microbe can grow in a medium where potassium cyanide is present as a carbon and nitrogen source. This test will also be used to differentiate E. coli from Citrobacter faundi species. In antibiotic susceptibility test, first we have the inoculum preparation. 5 well-isolated colonies will be selected from 18 to 24 hours agar slant culture. Each colony will be picked using an inoculating loop and then transferred to 15 ml with 3 replicates of lactose broth, then incubate for 18 to 24 hours at 30 degrees to 37 degrees Celsius. After incubation, 3 treatments will prepare the culture without any antibiotics. The second is a culture with streptomycin, and then a culture with amoxicillin, and these 3 should be done in triplicate. And then the second, we have the preparation of antibiotics. Streptomycin and amoxicillin antibiotics will be used in this study. Streptomycin will be labeled as antibiotic A and amoxicillin as antibiotic B, and prepare according to the recommended concentration. Third one, we have pipetting of inoculum and antibiotics on the optical density plates. There will be a control tubes and each column of the plate consists of three replicates for the control, for a culture with streptomycin and for a culture with amoxicillin respectively. The next one is the reading of well plate by microplate reader. The well optical density plate will be then read by a microplate reader with a wavelength of 600 nanometers. And then the last step or process that is also very important is the contamination. They use plates and tubes with growth after examination will be disposed and placed into a polypropylene autoclave bag, and then it should be tied with rubber band, and then steam heat sterilization will then follow in an autoclave with temperature to 100 degrees Celsius at 30 minutes. So I think that would be all. Thank you for listening.